All right. Hey, everyone. Once again, time for another rousing. Oh, my hair is all messed up. For another rousing rendition of physiology exam review. You guys have the review study guide, so I'm just going to kind of go through each point and then go ahead and record that, and you guys can take a look at it. Tomorrow, I'm going to have office hours. Actually, I incorrectly told you the time. It's going to be 12 to 1 and then 3:30 to 4:30. So that'll be the time. So, all right. So let's go ahead with the review. All right. So lecture exam 3 study guide. So 2 parts, obviously cardiovascular, which is really divided up into 3, right? We focused on the heart itself, ways of measuring, functions of it, blood flow, things like that, cardiac output. We spoke about blood itself, and then we spoke about blood vessels. So I'm just going to kind of go through briefly each of these just to kind of familiarize you once again with them. So blood flow through the systemic and pulmonary circuit. What you should know is some of those diagrams showing blood, how it moves through the body. The heart chambers, remember, just for example, for the systemic circuit, remember blood comes from the, ultimately from the pulmonary veins, which is oxygen rich, into the left atria, through the bicuspid valves, into the left ventricle, out through the aortic valve, through the aorta, and so on. So just know the blood flow patterns, which vessels, which chambers, and which valves. Speaking about the heart, we spoke about the heart function itself, and it's a pump, and it pumps in an alternating fashion of contraction or systole, where it's ejecting blood, and diastole, where it's filling with blood. This is known as the cardiac cycle. There's 5 stages to the cardiac cycle we spoke about, and what you need to know from them, excuse me, it's not that, but what dictates the different paths. For example, when the pressures are higher in the atria versus the ventricles, when the pressure is higher in the ventricles than the aorta, for example, in that case, blood is going to be ejected. When the valves are open, when the valves are closed. So pressures and within each stage, as well as the valve opening and closing, kind of like an input-output. Measuring cardiac function, we spoke about different ways of measuring it, things like the stroke volume. You should know the definitions. Stroke volume, remember the volume of blood ejected from each ventricle per beat. We spoke about the heart rate, beats per minute, cardiac output, obviously, and how EDV and ESV fits into the equation of stroke volume. There's a property of the heart known as which abides by the Frank-Starling law, which looks at the relationship between the EDV, or the filling time, or you can also call the stretching of the heart, thinking like a water balloon, and how much blood's going to be pumped out, or in other words, the stroke volume, filling time versus how much blood's being pumped out. You should know that relationship, and you should know, of course, with the term positive inotropic effect, with the sympathetic means that's really referring to, of course, is that the sympathetic branch of the autonomic nervous system increases stroke volume, which ultimately will increase cardiac output. Speaking of cardiac output, we looked at some of the factors. Remember, this is the volume of blood ejected from each ventricle per minute. We looked at some of the factors that influence it, namely the autonomic nervous system. You should know how each branch affects it, which affects stroke volume, which affects heart rate. We spoke about venous return, namely, obviously the factors that increase the return of blood back to the heart, which is important or essential, because the more blood you've got coming back to the heart will allow more blood to be pumped from it. So the different factors, respiratory pump, things like that. Heart being a muscle, myocardium, of course, has a mechanism of contraction, so you should know that. Remember, it's not neurogenic, it's myogenic, involving the role of the pacemaker cells, sending electrical signals through those gap junctions or intercalated discs. And you should know again, the calcium-induced calcium release. Very similar, most of it, to skeletal muscle. Of course, the difference is it's not neurogenic. With skeletal muscle, there's only one source of calcium. Cardiac muscle, there's two. So be able to kind of recognize the similarities and differences. I mentioned the pacemaker cells, which start the contraction of the heart. This is a part of a much larger group called the cardiac conduction system. You should know the parts of it and the order in which they're activated. The SA node, obviously, internodal pathway, AV node, and so on. Even though there's a built-in rate of contraction, built-in rate of activity of the SA nodes, it is influenced, of course, by the autonomic nervous system, so you should know how it's influenced. Remember, we spoke about depolarization, hyperpolarization. So you should know what the action potential looks like in an SA node, what an action potential looks like in the myocardium or the muscle, the significance of a refractory period. We also spoke about the heart itself. You know, there's many things that can go wrong. One of the things we spoke about is a condition known as murmurs, which is really abnormal sounds in the heart. So a physician can tell this by a stethoscope. Some of these abnormal flow patterns can be caused by valve stenosis, and we spoke about that. Remember, that's basically it doesn't open properly, fails to open completely, so that impairs the forward blood flow. So we spoke about what some of the consequences of that are. We also spoke about septal defects, right? The septum, of course, is a boundary between the left and right sides of the heart, and if that's breached, such as in the septal defect, that allows a mixing of oxygen-rich and oxygen-poor blood, which can affect our overall well-being. We spoke about blood plasma. We spoke about the blood, I should say. I'm sorry. We spoke about the blood itself. It's composed of both plasma and cellular elements. You should know the composition of the plasma, what the cellular elements, what they are, what their functions are, how the cellular elements are formed. In other words, what the stem cell is, what path they go down, what are some of the chemical messengers, right? Erythropoietin, colony stimulating factor that control each pathway. Regarding one of the cellular elements, it's a red blood cell, right? Of course, you should know how all the structures are, or how their structure of it. Remember, the red blood cell doesn't have a nucleus, doesn't have any organelles, but it does have a lot of hemoglobin. What does hemoglobin look like? What's its structure? Obviously has this ability to bind to oxygen. Remember, all red blood cells, once they get to about 4 months, they die off. They become fragile. Even though the cells die, we do recycle some things, so you should know how we recycle hemoglobin. Speaking also of red blood cells, that's where blood typing comes in. So you should know the features of all the different blood types, which antigens, which antibodies. And we did speak about, of course, that condition known as erythroblastosis fetalis, how that enters into the picture as far as with the RH. I think I did something here screwed up. Oh, I didn't. Okay, I'm good. All right, then we started looking at blood vessels. First of all, blood vessel histology, much like, what's the difference between an artery and a vein, an arterial and a venial, capillaries, layers, right, functions. We did speak a little bit more about the role of venous return. Obviously, it's occurring in veins, and what strategies we do have to ensure blood returning from veins back to the heart occurs efficiently. mechanics of the respiratory versus the skeletal muscle pumps, and of course, the importance of those venous valves. Continuing with blood vessels, we then spoke about the capillaries. Remember, this is where the exchange occurs, effects of and how the movement of fluid or gases, nutrients, whatever, out of the capillaries occurs at one end, the arterial end. That's called filtration, whereas absorption, movement of materials, waste, CO2, from the tissues back to the capillaries, how that occurs, and the role of the different pressures, which is higher, which is lower. And of course, I mentioned there's kind of an imbalance of fluid uptake where more fluid enters into the capillaries than is absorbed into the blood. So there's an excess. Fortunately, that's taken up by the lymphatics. Some other factors of blood vessels, of course, is we spoke about one way we measure cardiovascular function is what's called the mean arterial pressure. And the components of that, namely cardiac output and resistance, right? The balance of those and how the autonomic nervous system influences those to increase or decreases in response to high pressure or low pressure. We spoke about blood flow measurements, specifically Puiseuil's law. Know the different parts of Puiseuil's law, the components, how they affect blood flow, the increase of pressure, gradient, how does that affect blood flow? The increase in viscosity, how does that affect blood flow? Things like that. Finally, hemostasis, which was a big one, right? Hemostasis is mechanism of blood clotting, how looking at the different parts of that, looking at clotting disorders as well. All right, moving along to the respiratory system. We started off with some terminology, what ventilation is, what respiration is, inhaling, exhaling. We looked at the big picture, and when we talk about breathing, we speak about the ability to respire or respiration, external respiration, what that is. Remember, that's between the lungs and the blood. And then internal, that's between the blood and the tissues. Know what goes on there, know the movement of the gases. Again, the pressure involvement. Then we got into the whole anatomy, right? All the different structures of the respiratory tract, what their function is. Remember the conducting zone versus the respiratory zone. How does the structure of the trachea differ from the structure of the bronchioles? How does that differ from the structure of the alveoli? Remember, that's where the gas exchange is. That's where you have those different cells. That's where you have the respiratory membrane. Remember what that's made up of. We did kind of repeat some things. We did talk again about pressure versus volume in relation to breathing. And we repeated that throughout the discussion that Boyle's law is what governs breathing, really, the inverse relationship between pressure and volume. And then, of course, we did speak about the mechanisms of breathing. I'm going to kind of jump ahead here. How, again, the role of which nerves, which muscles, the role of the chemoreceptors, the pons, and the medulla on breathing itself, what their roles are. Speaking of breathing, of course, one aspect that's very important in our ability to breathe is what we call lung compliance, the ease in which it can inflate. And that has to do in large part due to the elasticity of the lungs. If the lungs are not elastic, if they're a bit more, fibrous, like I mentioned, that can occur with pulmonary fibrosis, they're not going to inflate as easily. I also spoke about, so those are some factors that we need to maximize. Surface tension is already high in the lungs, but fortunately we normally produce a substance called surfactant. Remember the type 2 cells and the role of the law of Laplace. So you should know how that fits into the large versus small alveoli and again, that whole relationship of regulating surface tension by interfering with hydrogen bonds. Surfactant is very important. I mentioned that we don't start producing it to the 34th week of pregnancy, or so a fetus that's born prematurely before 34 weeks typically will have to be on a ventilator for a while, or at least some sort of assistance. We spoke about acute respiratory distress in adults. So mechanism of breathing, like I mentioned, is important. And again, to elaborate, which muscles, which brain regions in the medulla, which regions in the medulla are involved in the quiet versus forced. Remember what the DRG involved with quiet, what muscles are does that activate? The VRG involved with forced, what does that activate? Getting to the home stretch, we started looking in the final lecture more about oxygen transport, CO2 transport. That goes beyond. Oh yeah, I got more to go. Never mind. Effects of CO2 on ventilation, local control of ventilation on blood flow. Remember, that was the ability, if you have a bronchial that's constricted, how does that affect the alveolar function? Obviously, it's not going to work very well, so we have to divert blood to the other vessel. Hey, Rye. Measurements of spirometry, how we measure, for example, lung function, such as tidal volume, FEV, those different parameters. We spoke about pulmonary disorders, and again, this is in no particular order here. We spoke about obstructive versus restrictive, and which are the different conditions that are associated with each of those. What happens, what is the cause, and what are some of the symptoms? We then jumped into the last lecture, oxygen transport. How, again, oxygen is transported primarily by way of hemoglobin. We then started looking, and of course, hemoglobin, without hemoglobin, we would be able to transport only a fraction of the oxygen. What are some of the factors that influence oxygen transport? Remember I mentioned, yeah, you can come and say hi. Well, you can't see them, but you can just say hi. I'm doing a review. You can just say hi now.
What's up?
All right. I'm going to be done in about 5-10 minutes.
Oh, no one's online?
No, I'm doing it sort of, I'm recording it. It's all right. Don't worry about it. After, have some lunch. After lunch, we'll vacuum, get your room all fixed up.
Yeah, we'll both do that.
Yeah, and then we can go for the long walk with
Awesome.
Roxy.
Yeah.
All right.
Do we have any tuna left?
Any what?
Tuna.
I don't know.
Okay.
We got the salads.
Yeah.
All right.
Roxy, hi.
All right. All right. All right. See you guys. I mean, they're not on, but I'm recording it for them. So, yeah, so don't say anything like that. It's all right. All right. So we're back. Sorry about that. So oxygen transport by way of hemoglobin. We spoke about the oxyhemoglobin dissociation curve. Remember that with metabolically active tissues, we have what's called the Bohr effect, which is due to, again, elevated DPG, which, again, detaches from hemoglobin, or actually, which binds to hemoglobin, I should say, allows oxygen to be delivered. We have elevated CO2, we have elevated temperature, when low pH. All of these contribute to a decrease in affinity, which will cause a right shift, Bohr effect. And then we have an increase in affinity to the left shift called the Haldane effect, which occurs at the lungs. This is, again, the opposite of these conditions. We have low DPG, low CO2, low temp, high pH. Some of the illnesses that are associated with this, we've talked about hemoglobin pathologies that can affect binding ability, for example, especially with oxygen. We spoke about sickle cell and thalassemia. We also spoke about the role of carbon monoxide and how carbon monoxide, unfortunately, has a stronger binding affinity with hemoglobin than it has with oxygen does. So if carbon monoxide levels are elevated, that's going to preferentially bind to hemoglobin. It's going to displace the oxygen, and we're going to suffocate. CO2 transport. Remember, there's 3 different forms of CO2 transport, principally bicarbonate due to dissociation of carbonic acid. Second most, of course, is bound to hemoglobin, carbaminohemoglobin, whereas remember, carbon monoxide is carboxyhemoglobin. And then finally, we have just carbon dioxide in the plasma. Finally, acid-base balance. What are the causes of respiratory acidosis and alkalosis, which is caused by elevated CO2, which is caused by decreased CO2. All right. Thank you for your attention, and I'm going to have lunch. Goodbye now.